Hi, my name is Sergio Hernández Torralba. I am a PhD student at Politècnica University of Valencia, and I am going to talk about my study based on the development of laser light backscattering imaging technique to model enzyme process on vegetable foodstuff matrices. Laser light backscattering imaging technique is based on the interaction between the laser light and food matrix. When the laser light comes into contact with a food matrix, a light pattern was formed. This pattern is recorded by a camera. These patterns and descriptors provide physicochemical information about foodstuff matrices and their transformations. Now, this technique has been effectively applied for modeling different processes in agri-food sector, as those related to enzyme transformations. The main advantage of this technique is that it is a low-cost and non-destructive technology that permits monitoring in a continuous way. The study This technique was applied to model enzyme transformations to soften vegetables. Carrots and pumpkins were chosen as vegetable solid matrices because of their elevated hardness. If not, it were suffered a prolonged thermal treatment that could be provoked the loss of vitamins and flavors, as well as organoleptic alterations in color and sweetness. Pumpkins were cut into cylinders of 2 centimeters of height and thickness, while carrots were cut into cylinders of 1 centimeter of height and 2.5 centimeters of diameter. Carrots were subject to pretreatment based on freezing and thawed to improve their porosity due to an expansion of their tissues as a consequence of a slow freezing and slow crystallization, and hence an improvement of their impregnation capacity. A commercial enzyme product with various pectinase activities was used. To improve the impregnation of the enzyme, vacuum pulse was applied. For this, samples were immersed into 0.5 enzyme solution and were subjected to vacuum pulse for 5 minutes. After that, atmosphere pressure was restored to complete the impregnation process. Carrots were suffered pulse in duplicate. After enzyme impregnation, samples were introduced on a dark chamber at room temperature in the case of pumpkin and at 36 degrees for carrots to accelerate enzyme reaction. Laser light backscattering imaging device was placed on the dark chamber. Light patterns generated on sample surface were recorded along the study. In relation to pumpkins, red point was used, while for carrots, red line was selected. The image processing and data acquisition for pumpkins was based on the calculation of exploring and quantification changes areas at pixel units. For this, RGB original stack was converted in 8-bit images with 255 values of gray to simplify the information. Then, the initial image of the stack was subtracted to all images of the stack with the aim to visualize only the pixels that changed. After that, the stack difference was thresholded to highlight the changed pixels. Finally, changed pixels were measured and plotted on the figure shown at the bottom of the screen. As can be observed, while enzyme-treated samples changed significantly, the control samples present a constant light pattern, as can be seen in the 3 images below from the initial time, at 110 minutes and at 200 minutes for the samples without enzyme. Moreover, as can be seen in the figure, most of the enzymatic transformations took place during the first 20 minutes. On the other hand, the images processing and data acquisition for carrots consists of measuring the increase of pixel value as shown indicated at the first figure. This region was selected because of its a central zone with the objective to determine if the impregnation at the center of sample was suitable and adequate. As before, the stack was converted at grade 8 bytes images, and now the increase of pixels values of gray was measured along the process. Once again, control samples did not present a significant changes, and the laser light pattern remained unchanged. However, as expected, enzyme-treated samples suffered a significantly changes. This evolution of gray values of the pixels was modeled following the equation shown below with a coefficient of determination of 0.98. Again, the most of changes were comprised at the first stage of enzyme process. In addition, the performance of changes led us to know that pixels varied from higher values. This suggests that increase the passage of light as structural deterioration increased due to the enzymatic action of the plant cell wall. As a conclusion, red laser point combined with the image processing method based on the calculation of pixels increase regarding the initial image permits determines the evolution of changes on the sample, which can be correlated with the physicochemical changes produced as a consequence of enzyme reaction. Moreover, it is possible to explore the region of the sample that suffer more changes. This fact provides information about the impregnation of the enzyme as well as kinetic aspects of the enzyme transformations. Additionally, the kinetic of the enzyme's reaction was modeled under an elevated coefficient of determination. The overall results demonstrated the capability of this technique for modeling different enzyme reaction and transformation process. This concludes the oral presentation of my poster. It only remains for me to thank the organization for the possibility to present my work and wish luck to the participants of the Congress.